Will not be a problem? No.
I just want you guys to feel comfortable. I mean, I'm recording just for Rashra. Hi, Rashra, because she's not here, and she asked for it. But I want, if the recording stays in the way for you asking questions, then I don't want that to happen, because now you have my time. Like, I barely have time to spend with people that amount of time. So now you have my time. So it's a good time to ask questions, right? Okay, so let me see. We do a lot of stuff in my lab. It's called the Living Matter Lab, and we work with living matter interfaces at Atlas Institute. Atlas Institute is part of CU Boulder, is part of the College of Engineering. I also am a professor in computer science in Atlas Institute and in biomedical engineering. So I span over 3 programs, institutes, and departments at CU Boulder. So what I do hardware, we do software, we do biology, and then we do art. And then we have an Instagram page where we post funny pictures from what's happening in the lab. So if you want to, like, you know, check out what we're doing from time to time and so on, it's an easy glance at the latest projects that we keep on doing. They are actually posted there quite in real time. So sometimes it takes months until they get actually published, or you get an official version of what's happening. So you can always kind of be in check with what they're doing, looking at the Instagram page. So I'm going to start with the fun things that I do in the lab and then go gradually more and more complicated, because Suresh said that, you know, he didn't understand a lot of things. So I'm trying to start with the things that are fun, and then I'm going to go towards the more complex and technical projects in our lab. So we have a lot of projects in art and design. Nada is a student that has a dance background. She's a research master student. She's going to apply and switch to a PhD next year. So she's working with glowing algae. These are what you see here. They are bioluminescent organisms that you can't see them. They're microscopic, quite big though, almost 1 millimeter. So you can the largest ones, you can see them. But during the daytime, you can't see them. And in the night, if you shake them vigorously, they start glowing. So we have an exhibition right now in Museum of Boulder, for those of you that are in town. We have I curated the whole exhibition in arts and technology, and one of the pieces is our piece on bioluminescent touch as well. So she's looking at how to interact directly with bioluminescence, because you have to shake it. It's a very embodied motion for that. So, you know, there are different environments. They have to be in liquid. That's one of the constraints, and they can be in porous surfaces. They can be open surface. So we build a bunch of games, right? Playing checkers, then playing Pong with them, hopscotch. And then we published a couple of papers on the topic. So that's one of the fun projects we have in the lab, and that I'm still working with them, keep working with different types of bacteria and different types of protists. These are protists. It's a class of organisms, and exploring various ways of designing with living matter. Again, if you're in town, check out the exhibition at Museum of Boulder. So Nada is doing the work that is at the intersection of livingness, organisms that are alive, microorganisms, and design and art. Fiona is one of my other PhD students. She's working with biomaterials. So I'm going to present 3 of her projects very quickly, very briefly. If you have questions, I'd love to go in detail and I'll answer them all. So one is Reclaim. It looks at food and takes food and transforms it into a clay, food compost. The other one is a self-cleaning textile, where we coated textiles with nano-coating particles, and we can program the way it cleans itself. It removes stains and bacteria. And third is a bioplastic that is edible, cheap, easy to make, and biodegrades in soil in 30 days. So Reclaim, this is taking compost, so your food waste, and composting it. It's a very personal material. And then we formulated a specific clay, and then we started working with it and see what we can do. And in general, we explore, as you can see here, this is drawn with a UV pen. So there's a UV reactive material, that pigment that we embedded inside the the clay. Here it's magnetic. These are small magnets. You can see this is in the dark. So this is fluorescent pigment that we embedded in it. We made a thermine, so we added some activated charcoal, which means that it becomes a capacitive sensor, and coupled with an Arduino, you can actually play music on it. Actually, we just got accepted last night, so I can show you the video. For every conference that we submit, we have to submit a video. And this is very recent work that we just learned it got accepted. So as you probably noticed, I'm trying to introduce it gradually. The first type of the project that Nata is working on on the design side is looking a lot at the impact. We are trying to have two different aspects in each of our projects, a technical part of it and an impact part of it. And that's more like, how does it transform our way of thinking of the world? So regardless of the technical difficulty, all the projects that are in my lab have to think of the impact that they have in the world. And in this case, Fiona is exploring biomaterials that are sustainable, that are degradable. So we're doing all this technical evaluation. You're going to see even more in the next projects. However, she's also looking at intimate making. So what does it mean that if the material is grown, if the material comes from your own food, if the material can be eaten, it's edible, how intimate is that connection with the material? And is that connected as well with sustainability? If you have to develop a more intimate material, are you going to like dispose it so often? Are you going to be taking more care of how you're using it? So the next project is a collaboration with a company called Accenture. That's a big software company, and they have just opened a research facility called Accenture Labs. So similar to Microsoft Research, similar to Google X, and you think of it. And then this is where my students usually do their internships over summer, and they are an excellent place for research. So in this case, we're working with textiles. The idea is to have textiles that are covering cinema seats and car seats in share rides that are able to have inside of them lights. And when the light is triggered, it starts cleaning the bacteria and the stains. And what you can see here are the experiments with the stains. So you can see the lights being on, and then you can see that the stain gets degraded, and we can even program the pattern we want into. It works in the sun as well. So this scrunchie was worn for 7 days in the sun, So then you can have different patterns. So this is called self-designing textile, and you can find a paper online. You can find a talk on YouTube on exactly how we develop that. Right now, I was actually just in the lab. I have a wet lab in engineering. A wet lab is a biological lab, bio-level safety one. And I was running the bacterial experiments on these textiles. So right now we are measuring how much of the bacteria we can kill by exposing the textiles to the light. And we are working with electronics and very tiny diodes, very tiny LEDs that are inside the fibers that make the textile. So cotton is a woven fiber. And we're hoping that it's going to become a commercial product that you can have on your car seats or in the cinema to clean them after they've been used. Then Algenil, this is a bioplastic that we just patented this summer. So it looks very much like vinyl, and it's edible. It's made of like agar, water, and glycerin. So we formulated this very specific recipe. Then we worked a lot with different pigments as well, similar how you saw with Reclaim. And then we tried to make clothing with it, and different other applications. So this is also like a very, very fun project that we have a patent on, and a couple of papers. You can see this is published recently in TI 2022, so we just got accepted for this one as well. And the Zinnia, it's a different journal, and the Reclaim is in CLI 2022. On the topic of my biomaterials, I also have a very fresh student. She just started 1 month, 2 months ago when the semester started, LD. She has since developed a biofoam that also degrades. The biodegrades in soil over 60 days. And this biofoam is supposed to be used for packaging. Let me see if I can find online the video on Atlas website, because this is a very new project. She managed to make, well, I think we can skip that. She managed to embed inside it fibers that are conductive. So that means that because it's a foam-like material, when you press it, the fibers get together, so the resistance changes, and that means it's becoming a resistance sensor. So it can be a smart packaging type, where if you have your electronics inside it and the box gets dropped and so on, then you can actually sense how much pressure has been applied, and how much a package has been traumatized. So this is a very new project. As I said, we're still working on it. We're hoping to publish in January, well, to submit a paper in January and publish in the summer on this work. Then I have two students. They are Latifa and Racy. They are both computer science students. All the other PhD students are part of the Atlas program. And then I'm also going to admit this year as part of the biomedical engineering program. So I can supervise students from multiple backgrounds and from multiple programs. So my lab is extremely interdisciplinary. Latifa and Racy kind of speak your language. They have a traditional, kind of a traditional CS background. Latifa is very CS. She's from Qatar. She did a master degree at UCL, University College London, and then Imperial College London, ICL. Well, yeah. And then Tracy has a master from Rice University, and his master's is in systems biology. So they are working in the lab right now on a project that combines electronics, computation, and biology, and microfluidics, a very interdisciplinary project. But the main living organism, because I kind of characterize the projects based on the living organism, is a phage. A bacteriophage is a tiny virus that attacks specific bacteria. So each bacteria has a very specific phage that eats it and attacks it. So you can see here the phage comes in the bacteria, invades it, and then just creates baby phages, and the bacteria gets destroyed. Now, why is this important? Well, it's important because the projections say that by 2050, more people are going to die of bacterial infections than of cancer. So because of the antibiotic resistance, which is called like the superbug problem, antibiotics are not effective anymore, but phages are effective. Phages are the natural enemies of bacteria. So trying to see, can we bring something called phage therapy on the market, and how can I use research to advance phage therapy? So we do that by using these devices called microfluidic biochips. What these devices do pretty much is that they move droplets around the surface of the electrodes, and I'm going to go extremely technical in a bit on how they work and what they do. They are electronic devices, so they're fully programmable. You see a bunch of electrodes here. We apply voltage from one electrode to another, and the droplet moves. The droplet can carry biological substances like bioreagents, viruses, bacteria, and so on. So what we're doing is that we're building the devices themselves, building the electronics, we're building the software that understands the bioprotocol, how to move those droplets and how to combine them, and then we are trying to replicate laboratory procedures on them. So this is one of the projects. This is called the phage box, and we're trying to amplify the phage DNA. The idea, the big idea behind it is, can we create new phages? Discovering phages in nature takes a lot of time. We actually have a class offered by MCDB, Molecular Cell and Development Biology here at CU, where students go out in the nature and try to hunt for new phages, try to discover new phages every single semester. There are 10 to the power 31 phages approximated in the world. So they are the largest number of organisms we know, and we know only about the DNA of about 5,000 of them. So there's a lot of work to discover new phages. They actually have to sample soil, they have to go through a lot of processes, and every year there's like 2 or 3 phages that comes out of CU Boulder, and they actually get to name them. So it's actually really fun. We're working in the lab with some phages that were discovered by our students here. So it's very exciting. What we're trying to do is, knowing that phages are quite simple as a DNA structure, can we use computation to find out what parts of the phages are important, and if we can alter them to mutate them artificially and create new phages. Instead of having people discover them in nature, can we replicate mutation and create new phages on these devices? So this is the big question that we're trying to answer. And one project that looks into it is trying to just replicate DNA. So you've heard of PCR. Now with COVID and everything, we're trying to take PCR and program it and automate it on these devices. Then another project, and this image I know doesn't tell you much to you, but it tells me a lot. We're trying to map proteins and figure out how to predict secondary structure of proteins. So it's a lot of computation behind it. It's only code. We've coded a lot there. Another project looks at, can we make phages fight each other by adding different types of bacteria in a phage cocktail, in a mix of phages, then sequence the data, take the sequencing data, and figure out if we can reconstruct the original phages. So, and this is a more complex problem than it seems, because the sequencing data that we get from a sequencer, so sequencer it just takes a bunch of DNA and outputs the A, C, T, G, all the nucleotides of the DNA. But the DNA comes mixed together, and the sequencer usually uses fragments and spits out everything in whatever order. So making sense of whatever data we get from the sequencer is an interesting computational problem. And with this project, we're trying to see if somebody goes to a phage clinic sick and not having any antibiotic that works, knowing that they will die in a month. And it's happening. There are people that are dying every day because of antibiotic resistance problem. The researchers there, they don't have too many options to find out the right phage for them. So they take their phages one by one and they try them on the patient. They try them in a Petri dish first for like 2 to 3 days, and then on the patient locally. So our idea is like, can we multiplex that? Can we mix all the phages together in a phage cocktail? This is what you have here. Then we add, here's the phage cocktail. Then we add the bacteria that we know that creates the infection to the phage cocktail. And because phages eat specific bacteria, we're hoping that the most infectious phage, the ones that likes that bacteria the most, is going to replicate a lot in our phage cocktail. So if we do that for a couple of days and then we sequence the DNA out, we're going to get an abundance of the DNA of the killer phage, of the actual phage that matters. So we're able to identify the phage faster than trying one by one sequential techniques. So this is another project in our lab that we're working on. Then we have another project called Phage 1. We're looking at a DNA of phages and at different functionalities of them. And we're creating, like we're using NLP, natural language processing methods, to create an insane amount of connected graphs and understand how many times in the same family of phages can we find this specific gene. And if we find it many times, and if it's located in specific areas, we know it's a very important gene. So we're looking at the structure of the phage and we're trying to identify what parts of the structure of the phages are so important that if we remove those, we know that we're going to kill the phage or we're going to affect its viability. So we did that computationally. We have a lot of code written and predictions on that. And now we're running experiments in the lab. We're testing our predictions. If we actually remove those parts, those nodes, because we created the genes, we model them as nodes, is the phage going to survive? Are our predictions right? And then we go and adjust our model. The idea behind this is to understand how phages connect and part of them together and what makes them mutate. So assuming that we want to create a new human from the existing humans, right? And we don't want to go through the usual reproductive way and, wait 9 months and get a baby and wait another 18 years until it gets mature. Assuming that we can replicate, we can clone people, but we want them to be still different. We want them to be their offsprings, not the exact clone of them. How would we do that? So we'll look at the structure of the human and we'll try to create exactly the difference that a baby would have by putting together two people, right? Two parents. So this is something that we do with phages. Phages don't need two parents to replicate. They replicate from the same, they mutate from the same phage, they create the same phage, so they're their own parent. So we're trying to look at the differences and how can we mutate and potentially create new phages. So these are a couple of aspects at high level. And before I go into detail into the biochip side, and I have a lot of slides on that and a lot of things, I want to stop for a second and ask if you have any questions. There are no questions. Okay, I'm going to switch to the technical presentation right now. Let me see. And I have a lot of slides here. Let me open the right one. Okay. So here, this is a biochip and this is how it works. So here's a droplet being moved on the surface of the electrode. This droplet is a very large droplet. It doesn't have a top. It has, the biochip doesn't have a top. It's a droplet of 20 microliters. So similar size with a rain droplet. That's quite large for us. We're usually working with way smaller than that. Now the idea behind it is, of course, behind using these devices is I would like to create this personal laboratory. So instead of going to the doctor, waiting to get the results back, what if you have a biochip in the doctor's office and you can get the results on the spot? And I love traveling. What if you have a biochip on you? Because one of the big issues when you're traveling is you don't have access to the healthcare. So this is what I call a personal laboratory. So the way you have a personal computer as your phone. What if you had a biochip that you could carry around and perform diagnosis and do a lot of things in a very interesting way? And right now I'm focused on the phage problems that I've told you before on these biochips, but this could potentially be expanded to a lot more applications than that. So I would like to create a generic medical platform based on biochips. Now, there are a couple of problems here to be tackled. We need to build a platform. It needs to be mobile, has networking capabilities. There are a lot of software aspects here. It needs to compile the bioprotocols from the natural language. So everything we do in the lab, everything that lab technicians do is expressed in natural language, and it needs to be compiled in machine learning code, machine code, sorry, not learning, machine code, so that, you know, the device can understand it. And then we have to automate the design of it. We have to have fault tolerance all the time because we're working with medical information. What about an editor for bioprotocols, or about an IDE for biology in general? There are questions about the service and application. And there's always the society part. Remember, I always think of the impact that we create by bringing the technology in this world. Who are the experts that are going to be using these machines? They could be potentially extremely, extremely powerful. And then what is the ethical problems behind it? And how can we embed that into the software and into a specific mode of use and mindset when using these devices? So this is one of, I built a lot of these devices. I have a couple of them with me here in my office. This is one of the first version that we released open source, so you can find it and replicate it. I developed it together with the do-it-yourself bio community in Europe. So a very, and probably here, Ibo is gonna laugh at us. The first version of it had a bunch of high volt transistors that we soldered manually. Now we got smarter and we have a transistor array actually there. This is a step-up converter that takes from 12 volts and goes up to 300 volts. You need between 120 to 300 volts DC to apply on these electrodes to move the droplet. You can also apply AC, but we prefer to do DC for this version because it was easier. And then the most complicated part is having a film on top of the electrodes. The traditional biochips, they usually have these very thin coats that are deposited very complicatedly in big machines like chemical vapor deposition. So we use a thin film like that you can use for the kitchen. This is like 10 microns and it has a red dielectric constant for it, and silicone oil as a hydrophobic layer on top of it. And then start removing droplets. As you can see here, a larger electrode array. You can mix droplets together here. With the top layer, we can split them as well. So what you see here is droplets with ink, so you can actually see them, because a lot of the chemicals we have in the lab, and here you have to believe me, are transparent. So, you know, here's DNA that I'm removing some bacterial DNA. Here's calcium chloride. We need it for genetic transformation. They look all the same. You really have to believe me, they don't have a color at this size. And this is ampicillin, an antibiotic we also use when it transforms genetically E. coli. So we also build like a very easy to use interface with drag and drop for people that don't know how to code Arduino, to just create new droplets, create new protocols, and have droplets mix and split. We have released this in 2017, and we've had since then more than 100 people replicated across the world and even fork it, because the design is open source. So I'm actually a board member in the Open Source Hardware Association. So we have MIT Media Lab built on a larger platform. They're using it for some LCD display. We have UW, University of Washington, using our original devices plus building another device for DNA computing. So they want to move DNA inside and try it there from a programming language background. So that's kind of their lab. So they're trying to see, can we do arithmetics and logics using DNA, and then actually the DNA being transported in these droplets. So these are my slides. I'm not going to go further into details unless you guys ask me or want me to. So if you have any questions regarding research, regarding how we do things in the lab, spaces, anything that I could answer, I'd be more than happy to take them right now. And again, we can also stop the recording if that makes you feel more comfortable.
Yeah, that does the work involve more of a hardware aspect, or like someone who's coming from just a computer science background?
That's a great question. So let me make clear, because I thought it's clear, but it's my fault. Let me make it more clear what is the software part here. I'm gonna share back my presentation, because it does seem like it's a lot of harder, and it is a lot of harder. So the big question is, where's the software? Here. The software comes into the compilation part. So this is how bioprotocols look like, right? This is what we do in the lab. How do you get from this one to moving droplets? This is what they called a compilation. It's similar to the compilers that you have in a computer, where you go and transform things into, depending the level that you want to go, up to bits, right? So what we do is that we model those natural language protocols as a graph, and then we use these graphs and then compile them and then store the results on a microcontroller, and that's how the droplets get controlled. So the compilation problem at computation level has a bunch of sub-problems. One is mapping each one of these operations, like dispensing reagents, for example, dispensing samples to the reservoirs on the chip. So you have the architecture as an input, you have the bioprotocol as an input. So you need to find which resources you need for each one of them. Some of them are reprogrammable. So for example, all these electrodes can be reprogrammed to do mixes or splits. So then you have another problem of choosing how to do the mix, because you can mix the droplets in many, many different ways depending on their viscosities. So that's a problem that's called mapping. Then you have to place those on the chips so that they don't intersect at the same time. They can intersect at different times because they can be reused after they're done. And then you have a scheduling problem. So how do you schedule them in time so that the resources are not coming, they're not overlapping, and at the same time the dependencies are respected, because you can't do this operation, for example, here before these operations are done. There's a dependency in the graph. So here's where we use a lot of coding. Actually, my students are coding constantly. And these are very interesting research problems. They're all of them NP hard, and some of them NP complete. So they're hard to solve. We are using different types of heuristics for it, from search heuristics to genetic algorithms to a simulated annealing based one, taboo search, ILP, integral linear programming. So yeah, there's a lot of interesting algorithmic side to this part here. It's a great question. Any other questions?
Yeah, I have a little question here. You said before that your project needs a lot of hardware design and hardware debugging, maybe. So I want to know some details about the hardware aspect,
Yeah.
and also
I don't have the images, the pictures from what is our latest hardware thing, because we just literally soldered it yesterday. But what we're trying to do is that we're trying to have everything that you have in the lab in the chip, right? And what you saw so far is just moving droplets, splitting them, so operating with droplets. But we wanted to have more. We wanted to have an incubator, for example, and we ended up using a TC component underneath that heats up. But we have to have that controlled. So we wanted to have like a region that gets hot and keeps the temperature at the right time. And we want to have multiple regions because PCR is actually cycling between temperatures. So we needed to have them at different temperatures. And we want to have an optical chamber where you can detect fluorescence, for example, on top of it, an optical module. Then we wanted to have a magnetic part as well, because you can use magnetic beads to collect the DNA. So we are constantly adding more and more things, because we would like to integrate all the needed components to run the bioprotocol from the beginning to the start. Because right now we're just moving droplets, mixing them there, and then we're taking them and going to like this other big machine and do stuff there. And this other big machine doesn't even know how to use how to work with such small amounts. So then we even like have to dilute it or add new things and so on. So constantly adding to the biochips and making them more and more functional and more integrate.
Okay. Thank you.
I mean, the big things there is that we're working with high voltage. Well, high voltage, we call it high voltage, it's not necessarily that high. So working with AC volts between 100 and 300 volts AC. And then the chips need to be programmable. We have them right now, the latest version, we have them on glass because we also want them to be autoclavable, so to be sterilized after. And as you know from chip design, if you design, we have like chromium electrodes deposited on glass, right?
Mhm.
So then it becomes also a question of designing the chip itself, like the electrodes, because you cannot have like vias, you don't have layering there.
Yeah.
But chromium allows for very thin wires. So you can
Mhm.
actually get wires very thin on the surface. So there are a couple of like very interesting challenges there from the design perspective. There's also a version of the chips that we don't have the expertise in the lab to work with, but if we get someone that is really good with electronics, there's called an active matrix biochip. They use thousands and thousands of electrodes. The way they build them is similar to LCD screens. So they use TFT transistors in the back, which means that they only use 20 volts because otherwise they just fry up. And that means that you can have like, instead of having reservoirs to dispense droplets, you can have like a large droplet there. You can manipulate them more fine and more granulated on those like large arrays. So that's something that we know it exists. We know that apart from the LCD technology, that is very hard to figure out because there's like a patent on it and so on, but it's kind of similar to LCD. I think somebody that has some background can actually do that very well. There's also some that are very similar as well that are called MEDA, MEDA biochips that use CMOS a base tech. So also hard to fabricate, but would be more than happy to support the fabrication part if we know somebody that actually can work with them very well. So check them out and look at the specs behind it and let me know how
Okay.
comfortable you feel
Okay.
with those
So as you said, I can say that the hardware design that are required in your project is mainly on the new function, right? Adding the new functions to your biochips, right?
Yeah, and we like also a larger array as well. But yeah, adding new functions, making them robust. I mean, it's not that easy.
So it's like an embedded system, right?
It is an embedded systems. Yeah.
And
My
it
PhD is in
also
embedded systems. Yeah.
and it also requires some programming on the basic algorithm. Yeah.
Yeah.
Okay.
Mhm.
Okay. Thank you.
But we do have, like, we work together a lot on this project, and we do have, really good coders in the lab. So if that's an issue, it's not, we can cover that part. We're lacking a little bit on the harder part. So the harder part, we're like learning as we go. I'm currently the most expert in hardware, which is never a good thing because I don't have too much time to solder. I have to do other things at this level. But yeah, my PhD is in embedded systems. So it is an embedded system. Yeah.
Okay. Okay. Thank you.
What if you have any questions? I'd love to hear them as well.
No, not really. Thank you. So,
Well, I don't want to keep you longer. Thank you for coming and bearing with me today. And if you have any questions regarding the program or the logistics, reach out to Raja. If you have any questions regarding the research, reach out to me. And then we're on campus if you want to see the labs. For those of you who are on campus, like the wet lab or the dry lab, I have a lab in a space in Atlas, another lab in the engineering center. Or if you just want to chat with the students in general, we're meeting. Our lab meeting is on Mondays at 4. So we're taking now the fall break, but we're going to be back after Mondays at 4 p.m. In Atlas, second floor. You can just find us there, and you can come and learn more if it's a better approach to actually see the people, meet them, and see the diversity in our group. It's a very diverse group, very different backgrounds. Fiona has a mechanical engineering background, Nata dance, LD design, Gracie biology, Latifa and Luis computer science. So we are used to being different and working together. Okay, I'm not gonna keep you longer. Thank you so much for today, and I hope to see some of you around here. Bye.
Thank you. Bye-bye.
Thanks a lot. I'm just gonna take off.
Mhm.